Good evening and welcome to an in-person Biotech 201. Whether you're joining us online or you are here in the Jackson Center, it is so nice to have you with us tonight. Thank you for being here. I've almost forgotten how to do this. So, somebody may need to remind me periodically if I suddenly wander off track or if I forget some of the key components. I know that we've already thrown you, those of you that are here in the room, because we didn't have cookies tonight, we had cupcakes. So, for those of you that are watching from home, tonight there was something totally different food-wise. We'll go back to cookies starting next week, but we had cupcakes and it was a real treat. So yes, you can applaud for the cookies because I know that that's an important part of your experience. As a reminder, we will not have an intermission. We'll go straight through, but if you need to step out and step to the back, please feel free to do so. I just want to start by thanking you for being here, for being with me tonight. I've missed you. I have missed this. I've missed us. And we wanted to make sure that we did not take any unnecessary risks. And I think that that was the wise decision. I'm glad that we waited a month. So thank you for shifting your schedules, your Tuesday nights along with me and with the Jackson Center. But I'm just really, really glad that we are here. And we have a really fantastic set of topics to cover over the next 4 weeks. So with that, why don't we go on ahead and get started? We are going to talk through a really cool topic, the science of gene editing. But before we jump into the science, I have a few words of thanks that I would like to give. I want to say thanks to the sponsors that have made Biotech 201 possible. Now, you have heard me talk about the James, Cecil, and Margaret Ashburn Foundation before. They have been longtime sponsors of Biotech 201, and I am deeply grateful. We are joined by a brand new sponsor this year, and I want to say a special word of thanks to founder Mark Spencer. So Avilution is our newest Biotech 201 sponsor, and Mark, thank you for allowing us to have tonight, so thank you very much. I also want to say thanks to the Jackson Center. Their support allows us to be in this incredible space, typically with our delicious cookies. And also to those of you that are Alumni Association members, your support also allows us to continue to offer Biotech 101 and 201. So here is the series. Here are the key points that we're going to cover tonight and the next 3 weeks. We're going to talk specifically about this idea of gene editing and what gene editing is. And so I want to make sure that we talk about the basic concepts behind gene editing. I want to also make sure that we grasp the applications. We're going to talk tonight just on what is gene editing and some early applications. We'll spend the next 2 weeks after that digging deep into how is gene editing being used? How might it be used in the future? We're also going to talk about some of the challenges. Gene editing is not the best thing since sliced bread. It is pretty amazing, but it does have some challenges and some problems, and it has some ethical issues. So I'm going to ask you to be thinking about how you feel about gene editing and about the impacts of this new technology. And then periodically, I'll also tell you how gene editing is being used on the HudsonAlpha campus as well. If you've got questions, I will probably pause at least once to take questions, but everybody should have some Post-it notes. Everybody who's in the room should have some Post-it notes. I'm going to ask you to write down your questions on those Post-it notes, and there's going to be a whiteboard as you walk out. Put those questions, put those Post-it notes on the whiteboard. We'll do this every week, and I'll gather those, and then I will answer those the following week. So it's a way for me to get a sense of what are you interested in? What are the questions that you've got? What are you wondering? If you are watching us online, put those questions in the chat. And our folks will actually gather those chat questions and they'll get them to me, and we'll do the same sort of thing. So lots of different ways for you to provide feedback, but I would encourage you to use the Post-it notes. If you don't have a pen, look to see if one of your neighbors has a pen. There are some pens in the back. If you still have a cupcake, you could maybe write with blue frosting if you needed to. But I do want your feedback. I want to know what you think and where you think this field might be going, and what excites you, and what potentially also gives you any concerns. So as I said, we're going to cover the foundations tonight. Next week we're going to talk about agriculture. The week after that we'll talk about human health. And then we're going to talk about some of the ethical concerns and challenges. Gene editing has revolutionized, and I don't use that word lightly, has revolutionized the field of genomics. And it has happened in an incredibly fast period of time. You are likely already eating foods from the grocery store that have undergone gene editing. There are people in clinical trials all over the world that are undergoing gene editing to treat their disease. There are lots of questions that still remain about what this field is and what the science potentially can and cannot do. So, any questions before we get started? Any general questions? You know, we haven't talked to each other in, 2 years since you last saw me in a wig and high heels. For those of you that don't know what I'm talking about, I was dressed as Louis the 16th. So that was where the wig and the high heels came from. Yes?
Yeah, I've got a friend who's trying to dial in or click in and he's not getting through. So I don't know if you're online, people can check and see if there's an issue.
Yes. Okay, so Stacy, if you can hear me, we've got somebody that's asked a question about being able to log in online. So if you can just check and make sure that the online link is working, we can go from there. We also video and record, and we'll make that available afterwards. So if for some reason we can't make the connection, they'll be able to view the video afterwards. Anybody else? All right, let's dive in. You may have noticed that I have on an apron. I did not make your cupcakes. And I have a set of different ingredients and a KitchenAid and a rolling pin, different utensils. I'd like to set the stage about what gene editing can be used for. So follow me on this journey for a second. I would like you to imagine that you have just purchased a worldwide restaurant chain that has thousands of restaurants all around the world. And you have an unbelievably broad menu. Every day, your restaurant makes hundreds and hundreds of different types of recipes and different foods for the people that come to eat at your restaurant. So there's a lot going on in the kitchen. The person that you bought this global chain from started with an original set of recipes that were curated over years. And then, as each franchise came on board, gave a copy of those recipes to the new franchise. And had a group of Benedictine monks hand copy the recipe book. So no copy machines. This individual was a bit of not a fan of modern technology. So every copy that went to each different franchise was hand copied. Now, I don't know if you've ever hand copied something, but you are likely to make some mistakes. A lot of mistakes, potentially, especially when you're transcribing things like recipes. Little t becomes big T, and teaspoon suddenly becomes tablespoon. 1/2 cup becomes 1 1/2 cups. Bake at 350 degrees is bake at 450 degrees. So those kinds of mistakes happen. And every single franchise around the world has the same cookbook, but does not have the same instructions inside the cookbook. Everybody with me on that? So you are now confronted with the fact that the recipes from Mexico City taste nothing like the recipe that you might find in Paris. And the recipes in Paris for coq au vin may taste really similar to coq au vin from the restaurant that's in London, but creme brulee may taste completely different. So no two franchises have the exact same set of recipes. They've got a lot of similarities, but everybody's got differences. And people in Tokyo hate the fried chicken, probably because it has 2 cups of salt in it. And people in Rome can't stand the brownies because they've been baked for 2 hours. All right, so there's your challenge. So you have all these restaurants all serving theoretically the same food, and many of them are serving the exact same things. But everywhere within those franchises, there's a little bit of difference. And every once in a while, people go wild about the change. They love the fact that there's chili powder in the chocolate pudding. Not a lot, but there's chili powder in the chocolate pudding. So you've got this challenge of, all right, how am I going to figure out with all of these global franchises what the best combination of recipes actually is? So what you want to do, I don't know if anybody's ever watched America's Test Kitchen. I love America's Test Kitchen. So these people go to town on how to fix your recipes. They change everything. They rework, they take it, they break it down and build it back up from the ground, and they make every single change you could imagine to find the best. You need to do America's Test Kitchen globally on every single recipe to figure out what really is the best way to serve lasagna or the best way to make chicken cacciatore. Then you also need to figure out, how do I now say, this is the ideal set of recipes? Everybody, this is going to be the new set of recipes that we're going to work with. And how do you also make sure that the things that say cook for 2 hours or 2 cups of salt get fixed as well? Everybody with me? That's the challenge, all right? So to put that into a different way, as the new owner, you have to optimize the recipes. You need to integrate all the best changes into all of the cookbooks around the globe, and you need to find and fix all the copying errors, especially those things like 2 cups of salt. So just try to imagine the challenge that you would need to do with that. Now I'm going to change the analogy. I'm going to take us to the world of science and health and farming. So now imagine, instead of the new owner, imagine that you're a scientist or you're a physician or you're a farmer. And you're working, maybe it's with a group of people, maybe it's with a group of plants, maybe it's with a group of livestock that at heart have the same cookbook, the same genetic code, but slightly different versions of the recipes. And some of those are the equivalent of 2 cups of salt. And some of those are the equivalent of, wow, that chili in the chocolate pudding is incredible. So you've got to figure out how you optimize the recipes, which is understanding how DNA change impacts the final function of the product. You've also got to figure out how you integrate the best changes. Now, maybe you're not trying to integrate the best changes into all the cookbooks, but let's say that you're a soybean farmer and you discover that there's a set of genetic changes that actually produces a soybean plant that has the best of all the different varieties of soybean. That it grows, it produces higher yielding beans, it grows under multiple different kinds of climate conditions, it's resistant to all the diseases that you would have to worry about. You would want quickly to bring that into the crop that you're working with. And you want to treat and prevent disease. If I know that an individual has cystic fibrosis or sickle cell anemia because they have this specific mutation, could I actually fix that recipe in that individual's cells? That concept, it is not an over-exaggeration for me to say gene editing offers the ability to do everything that we just talked about. It offers the ability to say what really happens if we change these recipes. If I target this specific change, what happens if I target every imaginable change? What do I ultimately get? And then how do I use that information to create varieties that meet the conditions that I'm trying to meet as a farmer or as a producer of sorghum or sugar cane? Or how can I actually think about treating disease? Now, some of you are thinking, wait, I don't want to optimize the entire cookbook. I really like some of the varieties, some of the recipes that are in that cookbook. And that's an appropriate question for us to ask. Should we be optimizing the recipes? Who decides what optimizing looks like? What if I don't want chili in my chocolate pudding? Who gets to make that call? And those are some of the ethical questions that come around gene editing. I think I am periodically cutting out on you guys. I apologize. So that's the power of what gene editing can do. I wanted to start with that analogy, with that concept of being able to make massive changes to recipes, optimize, and then think about how you make those changes on a more larger scale. I'm going to introduce the idea, and I'm not going to show you a video from QKED. It's a PBS station about gene editing. It's about 6 minutes long, but it introduces many of the themes that we're going to cover over the next 3 weeks. And it also does a really good job, better than if I had tried to create it myself. So in that spirit of someone else has already created the wheel, I don't need to recreate it, I'm going to step off the stage and we'll turn over to the video. And I think we'll have our sound ready just to make sure that it's not too loud. I'll be over here, so if you tell me that you need it louder or if it's too loud, we'll get it adjusted.
All right, here's a go-to sci-fi storyline that just keeps on giving. Scientists mess around with DNA, and then crazy things happen. You've got your classics like Jurassic Park. You've got The Rock's Rampage with a giant gorilla and a flying wolf with wings. But for me, I gotta go back to 1997 for Gattaca, where society uses genetic testing to create designer babies with ideal genes.
I've taken the liberty of eradicating any potentially prejudicial conditions, premature baldness, myopia, alcoholism, and addictive susceptibility, propensity for violence.
Late in 2018, the world of Gattaca took a small step into the real world. A Chinese researcher revealed that he created the first ever genetically edited babies. He altered the DNA of the twin girls before they were born, and what he says was an attempt to make them biologically resistant to HIV and AIDS. The world went kinda nuts, raising a huge bioethical question. When is gene editing okay? How far is too far when altering life itself? We can't talk about gene editing without first talking about genes, which are kind of like the instructions for life. All living things have them. They determine most of your physical traits, like the color of your eyes, whether your hair is curly or straight, the density of your bones, even how your heart responds to exercise. For thousands of years, humans have been unknowingly exploiting this in plants and animals through selective breeding. Every time a farmer decides to plant seeds from the biggest, juiciest tomatoes in order to get bigger, juicier tomatoes, they were indirectly influencing genes. Selective breeding is why we have hundreds of different breeds of dogs. I mean, come on, Chihuahuas and Great Danes are both the same species. Now that's crazy, that's a super wide spectrum. Selective breeding works, but it's imprecise and it takes generations before you see results. What if instead you could directly manipulate individual genes? That's exactly what gene editing does. The technology has been around. It's only been in the last 5 or 6 years that gene editing has really taken off. That's because of CRISPR, one of the biggest scientific breakthroughs in decades. It's a gene editing tool that can target specific genes, allowing scientists to make precise edits, much like a software engineer modifies code. They can cut out a gene, they can modify a gene, they can add an entirely new gene. If you want to learn more about it, our friends at It's Okay to Be Smart did a great video and you should definitely check it out. Now, every week there's news of another CRISPR breakthrough. One company is trying to turn pigs into organ donors for humans, using CRISPR so that the human body doesn't reject foreign tissue. A team of researchers in Texas a disease that causes severe muscle loss. CRISPR essentially reversed the muscle loss, creating some pretty jacked beagles. There are even scientists using CRISPR to add genes from the long extinct woolly mammoth into its closest living relative, the Asian elephant. They're basically trying to create a woolly mammoth elephant hybrid that might be able to better tolerate climate change. Now that's some pretty Jurassic Park level science if you ask me. But just because CRISPR might be able to do all this stuff one day, does that mean we should? If CRISPR delivers even a fraction of what we've been promised, it could be a game changer in how we fight diseases. Take sickle cell disease, for example, where there aren't enough healthy red blood cells to carry oxygen throughout the body. It affects about 100,000 Americans, and it's caused by a mutation in one gene among the roughly 20,000 found in humans. In fact, there are over 10,000 human disorders that are caused by single gene mutations. What if CRISPR could cure all of them simply by fixing the mutation? CRISPR could also help prevent infectious diseases. Researchers have been able to disrupt the reproductive cycle of mosquitoes, meaning they die Zika, and other deadly viruses that kill hundreds of thousands of people, many of them children, worldwide every year. If you want to learn more about that, we did an episode on it, and you should check it out. But things get really interesting when you think about how gene editing could affect multiple generations of humans. See, up until now, gene editing in humans has almost always been restricted to altering the genes in somatic cells, which are the cells in the body that are not involved in reproduction. So, any changes to DNA stay in that person. They can't be passed on. The Chinese researcher who gene edited those twins did something different. He did his CRISPRing when the twins were still embryos, meaning the reproductive cells were altered. So, any changes can be passed down to future offspring. Now, that could be great. If the researcher was successful in introducing a biological resistance to HIV and AIDS, then maybe we can one day get to a place where we can cure a disease in one generation and it disappears for all future generations. Now, as an astute above the noise viewer, you're probably thinking what I'm thinking right now. All of this sounds too good to be true. What's the catch? Well, while CRISPR is a relatively simple tool, the effects of altering genes might not be. Scientists have recently learned that CRISPR has the potential to wipe out or rearrange large chunks of DNA, which could do anything from turning off the wrong genes to triggering some forms of cancer. Also, who decides what's considered a problem that needs to be fixed? Sure, most of us might be fine with eliminating some diseases, but what about things like deafness or dwarfism or ADD? Many people in those communities see their differences as positive and adding to human diversity, not as a problem that needs to be eradicated. And even if scientists can perfect gene editing, which is a big if, a Gattaca-style designer baby scenario isn't hard to imagine. The rich and powerful can shell out the big bucks to customize their kids, protecting them from a bunch of diseases, reducing their risk for anxiety and depression, and maybe even giving them a heightened IQ boost. Meanwhile, the rest of us might only be able to afford a more limited amount of gene changes, if any. But what do you all think? When is gene editing okay? When should we use it? And when should we not? Let us know in the comments below. If you're a middle or high school teacher.
All right, so that 6-minute overview, which covered a lot of ground really quickly, I know, gives you a sense of the opportunity and the challenge around the concept of gene editing. Who gets to make decisions about what genes get edited? Now, the designer baby piece gets brought out every time somebody talks about gene editing. It is like the common thing that we're going to wave around. We're going to make designer babies and they're going to be 6 feet tall with blonde hair, blue eyes. They're going to be incredibly smart. They're going to have great ability to play basketball. Oh, they also have perfect pitch and they're going to make perfect scores on their SATs. Let me just take a step back and say that that concept gets pulled to the extreme a lot. Most of those things that I just talked about are not controlled by single genes, but by hundreds of genes, plus a really significant environmental impact as well. And so the likelihood that we're going to be able to designer baby is really challenging. I'm not saying that there aren't ethical issues around how do we make decisions about who has and who has not and who has access to those things. And we'll dig more into that when we get to our last week when we talk about ethics. Before I came to HudsonAlpha in 2006, I taught at Emory. I taught in the medical school. I taught first-year medical students medical genetics. And the very first day of every medical genetics class, I showed clips from Gattaca, from that very first movie that you saw. And at that point in time, it was really easy to say, we are nowhere near any of those things. It's a little less easy to say that now, because we actually can do in utero gene editing, and should we? And we'll talk about that Chinese scientist who in utero edited the genome of these twins, actually edited them at the embryonic stage before they were implanted, before they even became in utero, when they were in vitro in a dish. And we'll talk about that science as well. So we'll dig into where that field takes you. If part of you is saying, this could be an incredible way to treat disease, you are right. And if part of you is saying, I don't know how I feel about just willy-nilly changing things and like bringing back the woolly mammoth by breeding it with an elephant, you would be right as well. So we'll work through some of those pieces. But let's talk about what gene editing is at its heart. Gene editing is permanently modifying DNA at a specific site, and that involves two things. Permanently modifying means that I need to have some kind of system that when I get to the site, I can actually edit it, I can cut it, I can replace it. So I have some sort of, we often talk about these as like molecular scissors. I have some way to prep my DNA. And then at a specific site means I have to have a recognition system so I can find that specific site. The equivalent of go to the lasagna recipe and change the amount of ground beef and ground pork. That's being able to find the specific site. We're going to do a tiny bit of history, but not much, I promise. Gene editing really first took off in the early 2000s with the concept of something called zinc fingers. We're not going to spend a lot of time on this, but what I want you to see in this picture, here is the DNA sequence. And remember, the instructions that provide meaning are the rungs of that twisted ladder. This thing in blue and in red, these are zinc finger nucleases. So these are the parts that cut DNA. This is the cut-replace system. These, they look like rods or columns. This actually is the recognition system that says, here's the sequence that you're looking for. You can design these zinc fingers in a way to recognize a specific three-letter sequence of DNA. So this zinc finger recognizes AAT, and this zinc finger recognizes CGA. And if you put 4 of them together, you can now recognize a 12-letter piece of information. And if you put another 4 of them together over here, you recognize a different 12-letter piece of information. So it's a way to skim through your recipe and say, at this sequence, we're going to now cut. Zinc finger nucleases were unbelievably difficult. They were incredibly expensive, and you would spend years trying to get them to work. So in 2009, a group came up with something totally different called TALENs. And I don't even remember what TALENs stands for. This is one of those places where I'm now thinking, I really should have looked that up before I came up on stage tonight. I'm just going to chalk that up to being slightly rusty at doing in-person Biotech 201. But TALENs also gave you the ability to bind a specific sequence of DNA and to make cuts. And people got so excited about, well, I should say scientists. You probably never heard of TALENs. I shouldn't say people got so excited. Scientists got so excited. This was the 2012 breakthrough of the year in science, according to Science magazine. You probably all missed that entirely. But it was a big deal because suddenly there was the reality of really editing DNA. The intriguing thing is that about 4 months before TALEN became named the 2012 Breakthrough of the Year, these two ladies discovered what a completely different gene editing system. Actually, I should say they explained, because we already had seen it, but they explained and showed how to use a different system called CRISPR. Most of what we're going to talk about is around this field of CRISPR. And this story is really interesting. This is Jennifer Doudna and this is Emmanuel Charpentier. And the two of them, one from France, one working here in the United States in California, they were both studying this really intriguing system that happened in bacteria and thinking, I wonder if there is some way that we could actually utilize this system for ourselves. They did not set out to try to create a gene editing system. They were trying to think about how you could use this for immunity, and you'll understand why I say that in just a minute. They were both at a meeting. I think the meeting was in like São Paulo, Brazil, and they were hoping to meet each other, and they were introduced by a colleague in the coffee shop during a break in the meeting. One of the reasons why in-person meetings actually is really valuable in science, because virtually it's really hard to bump into somebody at the coffee shop when you're on Zoom. And they spent the rest of the afternoon having conversations about this, about how could they do this, what could this look like. They began to build a collaboration around this whole field. Now, here's the thing that I find fascinating. This work was done in 2012. These women won the Nobel Prize in 2020, 8 years. That's like a nanosecond. That never happens, but that tells you something about how powerful this technology is. So they both received the Nobel Prize in Chemistry in 2020. And if you want to understand this a little bit more, if you want to hear some of this in their own words, I would point you to the Nobel Prize website, that walks you through this. I'm going to show you some videos, and we'll pull some clips from one of those videos where Jennifer Doudna is actually talking about the science. I also would encourage you to read this book, A Crack in Creation. You can get this at Amazon. I'm sure you can get it in an ebook form as well. Jennifer Doudna, it's her telling of this process. And if you're thinking, oh my gosh, Neil, you tell me that you actually read this all about CRISPR and gene editing? I mean, is this like for insomnia? So Siddhartha Mukherjee, who also is a science writer, urgent, riveting, and endlessly fascinating. And some of you were like, wait, what? Riveting and endlessly fascinating? Well, actually, they do a really good job not only of talking about the science and the race for the science, but then the challenge that came of who gets to actually claim intellectual property on this, because that's where the big bucks are, and then all the different ways that this theoretically could be used and the ethical challenges around it. So if you find that this is really interesting and you want to better understand it, I would encourage you to get A Crack in Creation. It truly is a readable book. It is not like your high school science textbook, I promise. All right, I'm going to start with what CRISPR does in the real world, because the CRISPR that we're going to talk about for the rest of the time together has been modified to be able to do, let's optimize these recipes, let's find these changes, let's make these changes. The original CRISPR is an ancestral immune system for bacteria. So crazy that bacteria have a way to guard themselves against infection from viruses. But I want you to hear it in Jennifer Doudna's words. So she's going to tell you what the CRISPR system as it's originally intended to do, what it does, and then I'm going to talk through that and talk about how it's been modified to now be this unbelievably amazing set of molecular scissors. I need a second to get to the right place, because this is not the Nobel Prize video lets you link to it, but you can't clip to the specific part you want. So give me just a second.
Jennifer Doudna was born.
This video shows the way that we imagine CRISPR operates in nature, where bacteria that might be growing in a biofilm are being infected by viruses. And when the virus injects its DNA into the cell can acquire a small piece of that foreign DNA into the CRISPR locus, a place in the genome that consists of repeated sequences that flank these integrated sequences from viruses that are called spacers. And the cell then makes a copy of that integrated sequence in the form of RNA, which is then processed into individual units that include a sequence derived from a virus. Those RNAs combine with a second type of RNA called tracer and a protein called Cas9 to form a surveillance complex, an RNA-guided protein that can search the cell looking for sequences that match the sequence of the guide RNA. And when a match occurs, the CRISPR-Cas protein, Cas9, is able to cut double-stranded DNA, triggering destruction of the DNA in a bacterial cell.
So let's walk through that for just a second. Yeah, somebody said thank you. Yes, we will walk through that in a little bit more detail. So CRISPR was originally an immune response for bacteria. So I just want you to pause and think about that. Bacteria have an immune response. They have a way to guard against infection, because bacteria are continually being infected by tiny little viruses called bacteriophages. You and I at this point know a whole lot about viruses, don't we? more so than we ever wanted to, you know that a virus infects a cell and it injects its genetic material into the cell, and then that genetic material hijacks the cell's machinery and makes lots and lots of copies of that viral genetic information and the packaging component around it, packages it, and then sends it out to infect other cells. You know, we would have had to spend a lot of time talking about that 2.5 years ago. But we're all now pretty familiar with the ideas that viruses infect our cells and inject their genetic information. So here is the bacteriophage. Here's its genetic information. This is the bacterial DNA. And when a bacteria gets infected, it takes a tiny piece of the virus genetic information and it stores it in a library. You can think of it as a filing cabinet full of pictures of full of pictures of the viruses that have attacked it over the years. So, you know, it's like at the post office, you know, the most wanted set of pictures. Have you seen, you know, do you recognize these people? This is the bacteria's own recognition system of these viruses have infected me in the past. And it does that with this little tiny piece of DNA. And that is put in between these sections of blue DNA, which are the bacteria DNA, which are just repeating units. This is roughly the same set of information, ATTC, repeated over and over and over. CRISPR stands for clustered, regularly interspaced, short palindromic repeats. That's the only time I'm going to say that, I promise. But it's the only time I'm going to say it all together, but I'll break it down for you. Clustered, interspaced repeats. There are the repeats. They've all clustered together and they are interspaced. They're palindromic because some of them are flipped the other direction and they read the same way forwards as they do backwards. Madam, I'm Adam reads the same way forwards and backwards. A man, a plan, a canal, Panama reads the same way forwards and backwards. These repeated sequences read the same way, whatever orientation that they're in. So that's where the CRISPR name comes from. It's these sets of repeats. But the really important stuff isn't the repeats, it's the stuff that's in between. It's the viral most wanted lists. So this is in the bacteria's DNA. It's accumulated this set of, these are the viruses that I am looking out for because I have already been infected by them once. DNA makes a copy of itself into RNA, and that RNA in the bacteria gets processed. It gets broken up into little pieces. Here are the little pieces. Each one of them contains a little bit of that repeat and a little bit of the specific sequence of the virus. And then it combines, let's back up. It combines. Here's my RNA. It combines with another piece of RNA called tracer. We're not going to worry about what that means. And then it integrates in with a protein. So you've got a protein RNA complex. This protein is called Cas. And this protein is a set of molecular scissors. It chops up DNA. And it chops up DNA based on the sequence that it is given. So for example, now I've got all over my bacterial cell, I've got these little CRISPRs traveling around, these little Cas proteins traveling around, each with a little viral sequence. And it is looking at any genetic material that comes into the cell and saying, do I recognize you? Are you a virus I've seen before? Do you have the right similarity? Do I know who you are? That would be like if I was able to actually scan everybody in the room and say, I'm looking for Percy. And so I have a piece of information that matches Percy. It's Percy's picture. And it's looking at each of you and saying, are you Percy? And then it goes to Percy. Or it says, are you Helen? Are you Helen? Are you Helen? Ah, you're Helen. This is Helen over here. And when it finds Percy and when it finds Helen, when it finds a viral match, if it has been infected by a virus, it makes the match. So it says, yep, you're who this picture says I'm looking for. And then it chops the DNA. It cuts the DNA. So it is a find our match, chop. And that finding and chopping, when you cut the DNA of a virus, you actually trigger an entire system that breaks down the virus DNA and it destroys it before it can actually go any further. So it is a great way of saying, hey, is there anything here that potentially is going to do me harm? I've got a little bit of information about it. I'm looking. Oh, there it is. Chop. Okay, I've reduced my threat. Everybody with me on that? Do you have questions about that? Because that's a lot to process. I mean, we've only been here for 45 minutes after not being here at all for 2 years. So I know we've just covered a lot of ground. Questions around that? Yes, sir. Yeah, it's not like a what-cha-ha! Yeah, no, it is not that. So the Cas protein is actually called it's an enzyme called a nuclease, and the nuclease actually comes down around the DNA double helix and breaks the strand. So it actually is a cleaving process. It breaks the bond between two adjacent pieces of the DNA helix, and it does that. So your DNA has two strands, like the outside of your ladder, and the nuclease clips on this side and clips on this side. So it's a complete break. And when you have a break that occurs in DNA, DNA attempts to try to repair that break. And a lot of times it repairs it in such a poor way, if it doesn't have something to compare it against, that it just makes it completely, it no longer works. It just shuts down. And in the viral case, as I understand it, it actually triggers a breakdown of the entire virus genetic sequence.
Is this a function that most bacteria have, and do humans have anything like that?
Yeah, is this a function that most bacteria have, and do humans have anything like this? It is a function that you find in several bacteria, but not in all bacteria. And we don't, our immune system is more advanced than that. It recognizes, hey, I'm under attack. It ultimately learns the sequence of what is attacking it, and it generates an antibody response that then attacks the virus itself. I don't think that we have an equivalent, let's chop up the DNA kind of, well, we do have enzymes that chop up our DNA, but I don't think we have a CRISPR-ish system within us. Great question. Anybody else? Mark?
Don't we have a system that's designed to help battle cancer where it detects faults in the DNA and tries to fix it, and if not, triggers destruction?
So we have a DNA repair system that scans our own DNA and looks for breaks. And if it finds breaks, it tries to repair them. And if it decides it can't repair them, it initiates a cell breakdown, a cell self-destruction. So we do have that, but that's actually looking within our own genome. I'll actually go back and make sure, because there's a little bit of my brain that's saying, stop talking, Neil, because we actually do have some sort of system like that. Shut up, shut up, shut up. And so I'll actually go back and look for that, and I'll report back to you next week on that. There was another question. Yes?
I may have missed it, but back to basics. What causes that RNA to break down?
So there's an additional, it's a great question. So we've got that. And what is it that actually cleaves it into those individual pieces? There's a separate enzyme system, a separate enzyme that actually cuts that DNA within those repeated regions that breaks it down into individual pieces. So I believe that it is specific to how the CRISPR system works in bacteria. Anybody else? I have missed your questions. I've missed seeing you. I know I've already said that, but Percy, did you have a question?
Of course, did you mention what you were saying, talk about it, you mentioned pictures on the wall, as far as recognizing what if it's something new? Like now, with COVID, something new.
Yeah, what if it's something brand new that there's not a picture on the wall? Well, then that bacteria is out of luck. It's going to get infected. But it's going to pull a piece of that virus, and it is going to now add it to its list of photos on the wall. So it is a system that it continually can adapt. And every time that bacteria divides, remember bacteria divide by copying their DNA and dividing in half. It's simple cell division. The daughter cells that arise from that also now have contained that memory of what that of things that this lineage of bacteria has seen.
And what happens if it finds a piece that it thinks looks like that picture and destroys it, and that was something that
That's a problem. What happens if this system says, oh, look, you're that bad thing I'm looking for, and it happens to be a really important gene on its own that just had a lot of sequence similarity, a case of mistaken identity. That's a problem. Yeah. Unintended consequences. We would call that, and you'll hear me say this again, off-targeting. You're welcome. I'm glad you asked that. There's an extra cupcake for you. Now I'll get 60 questions. All right, so these researchers, Jennifer Doudna and Emmanuel Charpentier, were the first to show that this system could be utilized by scientists. It could be taken out of the bacteria and it could be modified to find and replace something else. That, I think, is the important piece here. There were lots of scientists before them that had tried to figure out, what is this CRISPR system? How does it work? But when they began to realize the specific mechanics, they said, hey, I think we could actually utilize this. We could take advantage of this ability to search and cut for our own purposes. And they were the first to actually show that. So this is the CRISPR version. This is that little piece of viral genetic information, and then this other RNA. These two scientists made a modification and created something that looks like this, and they called it the guide RNA. And that guide RNA is here in yellow, and this piece right here is modifiable, which means I could say, go find me Percy, go find me Helen, go find me David, go find me Nancy. I can tell it what to look for, what sequence to identify. Go find this specific sequence in this gene that regulates how many beans I get from a lima bean plant. Go find this sequence that is where there's a mutation that causes sickle cell anemia. I can target it to the sequence I want. And that is the power. I'm probably overplaying this, but the ability to tune what you're looking for is the key here. It was so difficult to do with zinc fingers and talons. It's almost ridiculously straightforward with CRISPR. And so that work was, yes, Hope.
I'm trying to go back and get it all together, but what the kid and the little once in a billion, is that how Jefferson Mill is?
No. So for that young man, Nick Volker, they didn't know where the mutation was, so they had to sequence his whole genome and find the change. They didn't know what they were looking for. CRISPR, you actually have to know, you need to know what you're looking for, or you need to design enough different guide RNAs that you've covered all of your bases. So it's a different technology. So they published in June of 2012. And at this point, I could tell you a whole bunch of scientific, and then they published in January of 2013, and they published right after that, and then they had already submitted an intellectual property, an IP application, but they had done it 3 days earlier, and who actually got to claim, who had the IP? At the end of the day, there are billions and billions of dollars in revenues at stake for this kind of tool and technology. And so the big question is, who gets to claim the revenues? And some of that's still working its way through the court system even 8 years later, because you're talking about billions and billions of dollars. And you've got places like the University of California and MIT and Broad that are willing to fight for what those dollars look like. So there's a lot of funding, there's a lot of money at stake, because this is now used all over everywhere. So two things can happen. I can cut the DNA, right here. I told you DNA doesn't like a break, does not like being untethered. So sometimes the whole gene just gets deleted. You just knock it out completely. But you could also take CRISPR and you could add another piece of genetic information that is a template that says, okay, when you cut this DNA, here's what I want you to put in its place. So that's the equivalent of go find the recipe that has 2 cups of salt and take it back to 2 teaspoons of salt. Go find the recipe that has 2 hours of baking time and take it to 20 minutes. So you can not just say, get rid of this, but you can actually say, and change it to that. And that's where the power also comes in. I can target it specifically, and I can direct the change that I want to make. Lots of people have improved on it. So you've got CRISPR 2.0 and CRISPR 3.0 and 4.5. It's like software upgrades. And there are all sorts of other tools and technologies that have kind of come along since then. But here's a challenge. We talked about off-targeting. If you have a sequence that's similar to what you're trying to find and cut, if it's really, really similar, you can unintentionally cut it and edit it as well. And that's a problem when you are making cuts in places that you weren't intending to make cuts in. So that was one of the really early problems of CRISPR, that you had all this off-targeting. In fact, one paper, if I'm remembering correctly, suggested that there could be 1 million off-target edits. That is really problematic. You are not going to try to treat somebody with a disease and then introduce 1 million new mutations. That would be like, all right, we're going to fix that 2 cups of salt, but I'm just going to now go change 50 other recipes at the same time. That does not help the process. But there are a number of new tools and technologies that have come along to actually try to minimize those off-target effects. They haven't made it 0, so this is still an issue. How do you make sure that you are not cutting in the wrong place? But it's much better than it used to be. So now I'm going to give you a preview of what we're going to cover in the next 2 weeks. These are all some headlines that I pulled just recently about how CRISPR is being used in agriculture. In 2018, in the summer of 2018, I think the first genetically edited oil, canola oil, hit the market and is available in the grocery stores. And it was edited to improve the oil profile and to make it last longer without going rancid. And that made its way through all the regulatory markets, and it was sold in the grocery stores. CRISPR is big business in ag. I'm going to introduce you to a bull named Cosmo next week. Cosmo has been genetically modified to increase the likelihood that he sires only sons, which is big business if you're in the cattle industry. Big business. So we're going to talk about that. We'll also talk about how some of the different ways that multiple plants are being looked at for gene editing. The week after that, we'll talk about the way that gene editing has taken therapies, clinical trials by storm. I'll introduce you to a young woman who was the first woman with sickle cell anemia to have her DNA edited and have the mutation repaired in her bone marrow cells. And in the 2.5 years since she had that CRISPR clinical trial, she has had no more sickle cell problems. Her doctor calls it a cure. That's a pretty big deal. That's a really big deal. But I'll also introduce you to the scientist from China who gene edited a set of twin girls to try to create gene edits that would prevent them from ever developing HIV. And there was off-targeting gene editing, and he didn't fully achieve what he wanted to, and the parents weren't quite sure what it was that they were signing approval for. So we'll talk a little bit about that as well. CRISPR has been used as a tool to rapidly detect COVID-19, as a rapid way to test, and when you find the viral DNA, you cleave it, and when you cleave it, you generate a signal that leads to a fluorescent protein. You heard in the very first video that researchers are looking at ways to genetically modify pigs so that their organs can be used in humans. You may have already seen this. This was at the very beginning of the year. This gentleman needed a heart transplant, was not eligible for a typical human heart for a number of reasons. We'll dive into that a little bit more, and got a pig heart. And that pig heart had been genetically edited to remove a set of genes that often cause, that are believed to cause rejection issues in humans. So they kept the immune system from over-responding and from him from rejecting it. This gentleman, they periodically show him back up in the news. I think that he was on the news a couple weeks ago because he watched the Super Bowl from his hospital bed. But anyway, he has a genetically edited pig heart that is keeping him alive. We will definitely talk about the ethical concerns around this. And I'll actually be seeking your feedback. So when we get to our last week, I'm going to be asking you a set of questions and asking you to say yes, no, maybe, I don't really know where I stand. And when we come in that week, I'm going to ask you to put some answers on some graphs so that we can get a sense of how people in this room feel now that we've spent some time digging into gene editing. I'll ask you, what do you think about the idea of gene editing for disease to reduce disease burden? What do you think about the idea of gene editing that doesn't just change your own cells, like your bone marrow cells, but actually changes all the cells in your body so that your children and your grandchildren inherit whatever change has been edited in you? On the one hand, that might be great because that reduces the burden of diseases in my kids and my grandkids. But what if we're not talking about disease reduction and who asked them if they wanted to have their DNA changed or not? That's a tricky kind of challenge. Who makes the decisions around what DNA should be edited and what should not be, and what is a disease, and what is just part of the diversity of human life? All right. I'm letting you out early tonight. We got to ease back in. There might even be some cupcakes out there still, but don't make a mad dash for the door. But next week we're going to talk about ag. We're going to dig into ag. So let me ask, do you have any more questions that you'd like to raise your hand and ask in this room? Anybody else? Mark.
Okay, sorry to be like asking twice.
No, it's okay.
But when we talk about the matching genomic sequence, how many base pairs are typically in that? Because I think of this as like you're editing a document, and the only thing you can do is do the find replace with the replace all option. So you got to be sure you type enough in the thing you're searching for, so that when you replace all, you're only really making the one change that you really wanted to make.
Yeah, so two things that I really appreciate. You just used my favorite analogy for CRISPR, which is find replace in our word processing, because that's exactly what it is. Find this sequence and do this thing to it. Delete it, edit it, put that comma here. I spelled that word wrong every single time in that document. Please fix it. But how do I make sure that I'm actually not off target? So there are different lengths of CRISPR guide RNAs. I want to say that they typically are in the 20 to 40 letter range, which is it's pretty specific that you're going to be able to get the string you want. But one of the things that scientists have to do, which is why it's important that we've got the genome sequenced of these organisms already, is they've got to go in and say, where does this sequence appear in any other place? And does it appear? So I know it appears in the gene I'm interested in, but does it appear in any other genes? And if it does, how do I then try to make sure that I've modified this in such a way that I'm only targeting that specific region being cut? Yeah, great question. Yes, sir. That's a beautiful question to ask, and I'm glad that you asked that. For those that might not have heard, the question was, what you've been talking about mostly in CRISPR is editing a single cell. How does that work when you're talking about editing a multi-celled organism? How do you edit that? So there are a couple of ways that you can begin to narrow down the number of cells that you have to edit. So for example, if you're trying to edit out sickle cell anemia, even though that mutation is in every single one of my cells, I'm only concerned about the cells where it's important that I have that mutation, and those are the cells that make my red blood cells. So I have to target my bone marrow. If you're talking about something that is specific to a liver disease, I only have to target my liver cells. So you first have to figure out what cell type you want to work with, and then how do you actually target, how do you home the CRISPR system? So you can deliver the CRISPR system, for example, in a couple ways. I could take a patient's bone marrow, I could take a bone marrow aspirate out, and in the lab, I can modify all of those bone marrow cells, and then destroy the patient's original bone marrow and give them essentially their own bone marrow transplant and let that grow back. So that'd be one way that I can target that. In the case of a liver disease, I actually can give the patient in an IV bag all of these CRISPR components, all packaged together in such a way that there's something on the outside of them that targets them to my liver cells. It's like a homing device just to my liver cells. So they'll circulate through my body, but they find their way to my liver, and they get into my liver cells, and they target just those liver cells. Now, are they going to target all my liver cells? No. But if I could get even 5% of my liver cells to now produce an enzyme that they weren't producing earlier, that's probably enough that my symptoms go away, or at least they get much less. If you're talking about how do I change all my cells, now we got to talk about I need to change all your cells when you don't have a whole lot of cells, i.e. when you are like a multi-celled conceptus or an early embryo, so that I can actually make those changes when there still is the potential for all those cells to grow and divide and become a fully formed human. So there are a lot of different levels to that question, and you're right, I skimmed over that, but how do you get the cells that you want is a key part of the, and so what do we actually do with this? Did that help? Perfect.
And what's HudsonAlpha's role in this field of science, gene editing? I mean, obviously, I know that DNA mapping and genome mapping is a big part of this, but kind of describe what we're doing here.
What are we doing in HudsonAlpha? I'll give you more details in the next couple of weeks, but essentially, one of the beautiful things about this tool is I can, for example, I can say, in this plant, how does this plant use fertilizer? What are the parts of the root cells that recognize when nitrogen is present in the soil, and how do they bring that nitrogen into the soil? What are all those genes? I can use CRISPR to silence those genes one at a time and say what happens. How does shutting that gene off impact how well this plant takes in fertilizer? Or this plant over here is a great utilizer of fertilizer, and it has this specific genetic change in this gene. Can I use CRISPR to take this plant that doesn't have that change and add that change and see what happens? Is this now the key? It comes back to, can I optimize those recipes? Can I read that instruction book more easily? So at the Institute, much of our work in CRISPR isn't, let's use this in a human therapy. It's at the earlier stage of, I can use CRISPR as a tool to make modifications back to America's test kitchen. I can modify these things and see, especially in plants, what that actually means, how that gets used. So that's a big part of that. We also are doing some work in cancer cells. If I make these modifications, how does that seem to impact how cancer cells in a cell line do? Does that give me more insight into ways you could think about new treatments? Yes, over here, and then I'll come right back here.
I work with people with disabilities and have for many years. There are some communities I know from the first few minutes they would hang me in ethics because they would do assessment. But help me understand, I'm an anthropologist. For this to work, the specific disease or genetic defect abnormality has to be identified
That's right. I should just hand the microphone to you. You've hit the key piece. That for us to think about treatment, number one, I have to know that this is caused by changes in DNA. And number two, I have to know what those changes are so that I can then edit them. A number of disorders do have genetic components, but the genetic component is only one piece of it. There are also environmental factors. So I could potentially do some editing to reduce risk, but I couldn't do editing necessarily to prevent disease. And you also touched on something so important. Who decides what should be treated? Who decides what is diseased and what needs to be fixed? And are members of those communities involved in the discussion? Or does someone just say, oh, look, this form of deafness has a genetic component, we're going to fix it, without asking if people who have this form of deafness feel that they need to be fixed? So who makes those decisions? And how do we engage people in those conversations? That's something that we talk about when we think about genetics and the stigma around genetic disease. So these are conversations we've had, but we need to now accelerate our ability to have these conversations, because this technology is not on your doorstep. It is in your living room watching TV and eating bags of Fritos. It's already here, and it is here to stay. So we've got to have some of these ethical conversations about how do we put some boundaries, how do we make decisions, how do we involve people, in these conversations. You get to be the last question for tonight. So, gene editing is not always done through bone marrow. If you're trying to treat a blood cell disease, you would do bone marrow. But if you have a disease where the mutation is important, let's say in your heart, doing something in your bone marrow isn't going to make a difference. You have to figure out how you're going to edit those heart cells. So you've got to know what specific tissue do you need to work with, do you need to be able to edit. And that comes back to the question from the back. How do I make sure that I get enough of those cells, I'm just going to keep moving around, enough of those cells edited that it makes a difference? I can't just fix one and say, okay, good, we're good to go. Now we're done. Okay, you may have other questions. You may have topics that you hope that I'm going to dive into. Use your Post-it notes. Take a minute, write on that Post-it note, and then put it on the whiteboard as you walk out. I will see you next Tuesday night. We will have cookies, I promise. For those of you watching online, put your questions in the chat, and we'll add those as well. Have a great night. Again, it's fantastic to see you. Thanks, everybody.